Hi guys, today I'm going to be describing the neurocontrol of skeletal muscle contraction. So first we'll talk about muscle fiber. This is a single muscle cell. It's composed of myofibrils, which are bundles of thick and thin filaments. The thick filaments contain myosin proteins, and then the thin filaments contain actin proteins. These filaments are organized into striated units called sarcomeres. Sarcomeres are the contractile unit of myofibrils. When stimulated, the filaments will slide past one another, overlapping, and this shortens the muscle fiber and causes contraction. The neuromuscular junction is the site where the motor neuron transmits an impulse to the skeletal muscle. It's a chemical synapse at the end of a motor neuron and a muscle fiber. It's composed of a motor neuron, which transmits the impulse to the muscle fiber, the motor implante, which is a part of the muscle fiber membrane called the sarcolemma, synaptic cleft, which is the gap separating the motor neuron and the muscle fiber membranes, synaptic vesicles, which store and release neurotransmitters, and the neurotransmitters are chemicals that stimulate or inhibit an effector. So stimulation of a muscle fiber begins with acetylcholine. This is the neurotransmitter that is responsible for muscle contraction. An action potential is conducted down the motor neuron, causing synaptic vesicles at the end of the axon to release acetylcholine into the synaptic cleft. Then acetylcholine will bind to receptors on the sarcolemma. This will increase the membrane permeability. Pretty much it will open sodium channels and close potassium channels, allowing more positively charged ions to flow into the cell. An action potential is generated in the muscle fiber, and it will be conducted throughout the muscle fiber. Once it reaches the sarcoplasmic reticulum, calcium channels will open, allowing calcium ions to diffuse into the cytosol. After calcium is released from the sarcoplasmic reticulum, it will bind with troponin, which is a protein on the actin filament. This will move the position of tropomyosin, which will expose the binding site on actin. This is where the myosin head will bind. A cross bridge is then formed between the myosin head and actin, linking the two filaments. Then ADP and phosphorus are released from myosin. This will cause the cross bridge to pull actin towards the center of the sarcomere. This is called a power stroke. The filaments will then overlap, which shortens the overall length of the sarcomere, causing a muscle contraction. For the filaments to go back to a resting state, first an ADP or ATP will attach to the myosin head, and the cross bridge will detach. Then ATP will split back into ADP and phosphorus, and the myosin head will be re-energized, cocking it back into its resting state. And here's my references.